Hey guys, so let's start our chapter now. Living world. Okay, so what is life and what are the defining features of life? In the NCERT textbook, they have given some. So let us discuss about them first and then go to the main points regarding the NEET exam. So, the defining features of life are cellular organization. What does this mean? That means if anything is living, it should at least have the cells. They might not be working together as tissues, but at least have a cellular organization. And then the next defining feature becomes metabolism. So what is metabolism? Metabolism is defined as the sum total of all the chemical reactions, more precisely the living chemical reactions inside the body. So the metabolism is directly linked to the cellular organization. Also, we can have a doubt that we can always perform a living reaction in a test tube, but the statement says all the living reactions occurring in a body. The living body doesn't mean any other body. I mean only the living bodies. Okay. I can refer the textbooks because I'm not teaching you, you're studying with me. And then conscious about surroundings. We are all always conscious about the surroundings. We can sense the environment. Here it's around 4:30 a.m. I can see that it's dark. I can sense the light. I can sense the temperature. Okay, not exactly, but I can say it if it's cold or hot, and maybe I can even change my body temperature. And that comes another point. That is self-regulating, which means that you can regulate your body conditions, the osmoregulations, the concentration of solutes in your body, the body fluids, and your temperature in the body. Okay, these are all the defining features. You can also include these things, interaction. These come under the defining features as well. Obviously, right? All the cells interact with each other to form tissues. The tissues interact with each other to form organs. In that way, it proceeds to the level of organism. Okay, these are defining features. So let's go to characteristic of living beings, but they are not necessarily the defining features, and we'll also know why aren't they the defining features of life. That is growth and reproduction. Growth. Okay, how do we define growth? Growth means it can be either of cell division, or by actual cell growth. For small organisms, cell growth means cell division. They grow, then they divide. That is method of reproduction as well, and the growth. So the living organisms grow by cell division and to an extent the cell growth. But why isn't this the defining feature of living organisms? It is because even the non-living things grow. See, as I keep layering this layer to micro extent, this thing is growing, right? Okay, it's not the same material, but if I keep adding on this white layer, it's on the white bowl, it will keep growing. It increases its length, thickness, so it's growing, but that is not living. So this is just a characteristic of living things, but not the defining ones. Another thing which is not a defining feature but is still a characteristic feature, that is reproduction. Under reproduction, almost all the living things reproduce and produce their new offspring. I said almost because there are some exceptions. For example, the male honeybees, drones, they are sterile, they did not reproduce. And infertile couples, they are living, right? They are living, but they cannot reproduce on their own. So these are called characteristic but not defining features of life. Note one more thing, this is important. All organisms have consciousness, but only human beings have self-consciousness. Another important statement, it's not given from the example, but it means a lot. It is all the living things are linked to each other by sharing the common genetic material, but to varying degrees. For example, so let's start with nomenclature or taxonomy. This term is given by A.P.D. Candolle. Remember this name, this is important. Taxonomy means the study of processes and principles of classification. And there are three types of taxonomy, that is alpha, beta. Okay, I'm not gonna write this. Shit. Alpha taxonomy considers only the morphological characteristics of the organisms, and the beta taxonomy considers all the important characteristics. And then comes the omega taxonomy, which includes all the important characteristics, just like the beta taxonomy, including the phylogenetics. So what is phylogenetics? Phylogenetics is based on the evolutionary sequence. Phylogenetics
based on evolutionary sequence.
And phylogenetic tree is also called a cladogram, and a phenetic tree is called the dendrogram. Okay, let's go to the processes of taxonomy now. Processes in, I hope you can understand my writing. First, characterize the organism. List out all the characteristics of the organism. And then identify what the matching characteristics to the already listed characteristics you have identified before. If that is new, congrats, you have discovered a new organism. Then classify it according to the identifying points. Once you have classified it, you can, if that is a new organism, you can give it your own nomenclature based on the nomenclature rules. These three are the scope of systematics. Scope of systematics. We'll get to that right now. So what's systematics? Systematics is the taxonomy plus phylogenetics, that is the evolutionary relationship we just studied. Systematic, like define what is systematic. It's like defined or regulated in an orderly manner. Taxonomy, it's the naming. These things are taxonomy. And in an evolutionary sequence, if you are characterizing, it's called the systematics. Evolutionary sequence means phylogenetics. There are various other taxonomy as well. It's like numerical taxonomy, chemotaxonomy. Numerical taxonomy is based on the observable characters, and numbers are code. Numbers and codes are assigned to all the characteristics, and then data is processed. Each character is given equal importance. It's not like one character is more priority, like flower is more priority than leaf. It's not like that. Each character is given equal importance, and hundreds of characters are considered at a time. It's a processing, right? It's mostly based on computers. They can't process everything. Chemotaxonomy uses cellular constituents and it just gets rid of the confusions. Let's go to nomenclature. It's the last process in taxonomy. So the rules for taxonomy, they are generally derived from Latin or in the Latinized form of English. From Latin or Latinized form of English. And if you are writing in hand like I am doing, you must underline it because you can't just italicize your writing, right? And if it is typed, you can italicize it. If handwritten, it should be underlined, and if typed, then it should be in italics. And next, in binomial nomenclature, which is given by Linnaeus, the specific epithet comes after the generic name. For example, Mangifera indica. You should note that I have written the generic name in capitals and the specific name in starting with the small letter or the lowercase letters. And that is also a rule now. It should be seen. The generic name should start with capital letter and the specific name should start with a lowercase letter. Also, another rule says the name of the author or the discoverer should be followed next. This is discovered by Linnaeus, so the full name but just the half name, just cut short name. So these are the rules for taxonomy. I hope I haven't missed anything. There are two nomenclatures now. Binomial type, which we just discussed. Binomial nomenclature. This is given by Linnaeus. It consists of genus plus species. Note, I have written this in capital and this is in lowercase. You should note that. For example, Mangifera indica. It's the mango, if you don't know. And next, trinomial nomenclature. This thing is given by Lamarck. The giraffe guy. You'll know what I meant in the evolution chapter. It consists of genus, species, and subspecies as well. For example, where should I write it? Corvus, it shouldn't be one after one, it should be in a straight line. See, this is the generic name, this is the species name, this is the subspecies name. It's the same for both, and the common name for this organism, Indian crow. Seeing this, it reminds me of tautonomy. So what is tautonomy? When the genus name and the species name are same, for example, Naja Naja, it's the cobra. Here, you should notice that genus name and species name are the same. Also, axis. This is spotted deer. This is called tautonomy, where both the species name and genus name are same. All the species concept, we'll just refer to that once. The biological species concept is given by Ernest Mayr. Biological, given by Ernest Mayr. He says that species are reproductively isolated from each other. In his concept of species, for example, these two organisms of the same species, they're not isolated reproductively, and after a long evolutionary time, this becomes a new species and this becomes a new species. Due to reproductive isolation, this may arise due to geographical isolation as well. And another species concept was given by Linnaeus, and that is morphological species concept, and he wrote all about that in his book Systema Naturae. I'll write about it later. Okay, let's go to taxonomical aids which help in identification of new species. First comes the herbarium or herbaria. It is a quick referral system in taxonomy. Note this point, this is important for NEET. Quick referral system in taxonomy, and just it's only for plants. Only for plants. The processes in herbarium. Collection. You have to collect the plant specimens specifically in vascular bags such as polythene bags to prevent the entry of moisture, and then you have to dry it, poison it to stop the fungal growth or any growth of any other microorganisms. Okay, the poisoning is done by is done by dipping it in 2% solution of alcoholic mercury chloride. Dipping in 2% solution of alcoholic mercury chloride and then mounting it on the 11.5 inches into 16.5 inches sheet and then labeling. Labeling is done in the lower right corner in here. If this is a sheet of paper, it's done in here, lower right corner. Botanical gardens. Contrasting to herbarium, it has the living plants for reference and identification purposes. For example, Royal Botanical Garden. It is in Kew, England. It is the botanical capital of the world. It has the largest botanical garden. Kew, England, Royal Botanical Garden. And next, in India, there is Indian Botanical Garden in Howrah, Kolkata. It's the largest in India.
Botanical Garden. It's in Howrah, Kolkata.
And this is the largest in India. And then National Botanical Research Institute, NBRI. It is in Lucknow. Maybe asked in exam, so it's important to note it down. And then museums. Museums have preserved plant and animal specimens for study and reference. And animal. They are preserved in preservative solutions such as formalin, that is 5% solution of formaldehyde, and then the larger mammals, birds, are stuffed and preserved, and insects are collected, killed, and are pinned onto boards. Mammals or birds. Stuffed and preserved, while the insects, they are collected, killed, and then pinned onto boards. Next, this is an important one, keys. Keys are the tools which help in identification of organisms based on similarities and dissimilarities. Similarities and similarities, and they are generally analytical in nature. And each statement in a key is called the lead. Each statement in a key is called a lead, and the similarities and dissimilarities are the contrasting characters. And each pair of contrasting character is called a couplet. And then manuals. Manuals contain information about only one area, and monographs contain information about any one taxon. Flora, it is the actual account of habitat and distribution of only plants of an area. Manual in an alphabetic order is a catalog. Okay, let's go for the bonus points of today. Father of taxonomy. Is Linnaeus. And then father of botany. Can you guess? Yeah, Theophrastus. Hope the spelling is correct. Father of biology is Aristotle. And then father of modern biology is also Linnaeus. Linnaeus. Let's also look at the books they wrote. Not only they, some of the most important books as well. Aristotle. It's the author, not the book. He wrote the book called Historia Animalia. And his student Theophrastus, he's the father of botany. He wrote Historia Plantarum. And then John Ray, he wrote about Historia Generalis Plantarum. And then Linnaeus, dear father of modern biology, botany, he wrote Systema Naturae. This book included the binomial nomenclature. Also, he wrote about Species Plantarum. In this book, he classified 5,900 species. 5900 species. Please note them down. These are important. Okay, sorry. Then you can always pause it, right? It's not a live class, man. You can pause it. Until now, we haven't discussed the order of classification. Okay. First comes the kingdom, and then phylum, or if it's a plant, it's called division, and then class, order, family, genus, and species. Okay. For example, we'll go as humans. We belong to the kingdom Animalia, phylum Chordata, class Mammalia, order Primata, family Hominidae, genus Homo, species Sapiens. Also, the subspecies is Sapiens. An important NCERT statement is given us. As we move up in the classification from species to the kingdom, diversity increases. Diversity increases, but common characteristics decrease. Decreases. This is an important thing. Okay. Here, before I end this session, I'll also tell you about some of the code names. The nomenclature and the rules of the nomenclature are given by the ICBN. That is ICBN, International Code for Botanical Nomenclature. This is for botany, and for zoology, ICZN, that is International Code for Zoological Nomenclature. And then ICNB, it is for bacteria, International Code for Nomenclature of Bacteria. And then ICNCP, it is the International Code for Nomenclature of Cultivated Plants. And then ICTV, this is International Code for Taxonomy of Viruses. That's it for today. Hope you all enjoyed the session, and do not forget to revise NCERT first. Thanks for watching.